heterospore is seed habit also. What is stellar evolution here? Means steel means steel means is the central core system, a pillar-like structure or a central core system is there. In the lower plants, what happened here from the Thallophyta to Bryophyta, the conductive of nutritive fluid takes place by the cell diffusion. There is no special type of the conducting tissue. Onwards, Pteridophyta onwards. Only the Thallophyta, Bryophyta, there is no special type of the connective tissue. So from the Pteridophyta onwards, there is a specific type of the connective tissue is there, that is the stellar or vascular system. So here, what happened here, the the sporophyte as the dominant generation, which has not only increased size but has also developed as a complex structure. So that is the differentiation. This vascular system is gradually developed. Okay. So the vascular system is appearance for the first time in the pteridophyta only. So that's why they are called as a vascular cryptogam. Pteridophyta or the cryptogam means there is no flower. So that's why the vascular cryptogams are there. So first of all, present the vascular system in the pteridophyta only. Okay. So now the older anatomists like Nageli and other supporting phytonic theories are there. They are the vascular stand of the leaf. They are the pushing to its course for the leaf to access as the structural unit of the vascular system of the pteridophyta. So, however, in the Vanthigam, so they are described the steel. Steel is a Greek word, means which is the pillar or connecting tissue or central core. It is the whole central mass of the vascular system with or without pit, means which is the extremely covered by the endodermis and fundamental unit of the vascular system. So this forms the base of the stellar theory. The stellar theory holds that the body of the system or root, system of stem or root looks like a, they are look alike with the base structure is constructing the central cylinder, shield surrounded by the cortex. Yesterday we know that one, we discussed the central core is there, which is surrounded by the cortex, and upper epidermis is there. So in the in the plant kingdom, the vascular system has its origin in the Pteridophyta, which is also called the first vascular plants. Which are we called the first vascular plants? These Pteridophyta members are called as the first vascular plants. So these pteridophytes are also considered the primitive vascular plants in course of the evolution in the simple and primitive vascular system. So some several progressive variations lead to the formation of the complex steel as they found in angiosperms. So the evolution of steel is studied from three aspects are there. What are there here? The types of primary xylem, composition of the xylem and phloem, another one the structure of steel. Here we can consider three aspects: types of the primary xylem and complex of the xylem and phloem. Another one is the structure of steel. So here types of primary xylem. Three types of the primary xylem are they are recognized in the relationship between the protoxylem and metaxylem with reference to their position. What is the protoxylem and what is the metaxylem based on their position. So the xylem may be differentiated from the procambium from the point of origin toward the center of the axis. So the xylem so formed as a result of the centripetal grow. Centripetal means outside to the center, periphery to the center, that is the centripetal. So it's called a centripetal xylem. So it's not audible. Okay, no clear. So in this, the protoxylem is situated at the away from the center of the axis and the metaxylem toward the center. So such primary xylem is called as the exarch. Means the protoxylem is away from the center of axis. That one we call as the exarch condition. Metaxylem is towards the center, means axis. That is the exarch condition is there. So the xylem may grow to the point of the beginning towards the periphery of the axis. This result formation of the centrifugal xylem have the protoxylem toward the center of the axis and metaxylem away from the center. Such xylem is known as the endark condition. Okay, the protoxylem is away from and the metaxylem is near to the center. That one is the called as the endark condition. Such xylem is known as the endark xylem. In certain both centripetal and centrifugal xylem is formed, such as the metaxylem occurs around the protoxylem. This one is called as the mesarc condition. Protoxylem and metaxylem. Metaxylem is occurs around the protoxylem. That one is the mesarc condition is there. So next one is the composition of the xylem and food. Xylem is the complex tissue. We know that one. Xylem is a complex tissue, so which is intended for the conducting water and minerals. In the pterophyte and gymnosperm, with a certain exception, xylem is consisting tracheids. Tracheids are there. In certain other additions, the tracheids they may be xylem parenchyma. And this is the tracheids are the very primitive character in the xylem. Okay. So here the phloem is a complex tissue, we know that one, which is conducting the food substances. Xylem is only to connect the water and substances, but here minerals. The phloem is a complex tissue which is connect the food, conducting food transport system. In the pterophyte and gymnosperm, the phloem consists only the sieve cells. This is the very primitive character. The presence of sieve tubes, companion cells, and phloem parenchyma is the advanced character. Is there. Only sieve cells are there in the pterophyte. So the companion cells are totally absent in the gymnosperms and pterophyta. So in the gymnosperms, like instead of the companion cells, the albuminous cells are there. So here I speak loudly only. The page number here is not audible. Hello? Hello? Is it audible?
Yes, sir.
So I speak loudly only. Some members are start with speak loudly like that. I always speak loudly. Okay. So what is there here? The companion cells are absent in the tetraphyte and gymnosperm. Instead of the companion cells, here gymnosperms, albuminous cells are there. Okay. The next one, the structure of steel. The structure of steel here, that is the evolution of steel basing on the structure of steel in Pteridophyta. The trace from the different sources are there. A comparative study of the structure of these steels is present in the living genera from the main source. second is the study of the structure of steel in fossil forms, and it is provided in the comparative study of ancient and modern pteridophytes. Okay, so first of all, we discuss the evidence. Uh, what is the evidences are there in the form of the fossils? So thirdly, the study of evident developed steel during the growth and individual also throws the light on the evolution. Steel is ontogeny. Ontogeny repeats phylogeny. During the evolution, the steel is pterophytes shows the progressive variation from the protosteel to as a polycyclic dictyosteel. From the variation means the diverse development from the protosteels to polycyclic dictyosteel. What is the diversity is there? Shows the progressive from the polysteel to polycyclic with the transitional stages, that is the siphonosteel, solonosteel, and dictyosteels are there. These are the middle intermediate steels are there. Protosteels to polycyclic dictosteel. This is very advanced steel. Polycyclic dictosteel is very developed condition. So first of all, we discuss the protosteel. So protosteel means only simple tissue, simple connective tissue is there. This is the simplest and phylogenetically primitive type of the steel. Very primitive structure in the protosteel, which is consist of the central solid core surrounded by the layer of phloem followed by the pericyclic medullosa. So here pith is absent, totally absent. Only the xylem is there. The xylem is surrounded by the phloem. This one is the haplosteel is nothing but protosteel. Okay. So here all other the types of steel have been derived from the course of evaluation. So this the starts with the protostelic system is very young stage. In the permanently retained, the adult stem is fossil as living Carotophyta, like that in the Rhinia Braptis. These are the protostele is there. So in this form, the leaf trace is the simple construction is a single vascular strand with the split from the protostele. So like this, only the single strand leaf traces. The leaf trace is leaves to break in the central cylinder. Okay. So with increased the demand for connecting the evolution, steel has taken place in resulting the variation of the protostele. In haplostele, so some variations are there in the protostele. That is, the haplostele is very smooth structure in the xylem is surrounded by the phloem. This one. And in the actinostele, yesterday we discussed the actinostele. You can see in the lycopodium. In the actinostele, the xylem is expanded by the developing radiating ribs with the protoxylem located the extremities, which is surrounded by the phloem. That is, the actin-like structures are there. The xylem is actin-like structures are there. Extending ribs, radiating ribs are there. This is the actinostele. And the actinostele has further undergoes the variation there to separate plates as they are arranged like this plectostele. A parallel plates like structures are there. That one we call as the plectostele in Lycopodium volbili, volbili, Lycopodium volbili. So already yesterday we discussed the actinostele, plectostele, mixed protostele, like that. Okay. So mixed protostele here. The mixed protostele means the xylem is breaks down into small patches. Patches like structures are there. That xylem is totally scattered in the phloem. That one is the mixed protostele. Okay. So next one is the siphonostele. What is the siphonostele? Means some species of Carotophides is the is large and thin-walled parenchyma cells may be associated with the xylem. That one is called as the pith. In Glycinia, is the large central core xylem is composed. The tract is interminate with a wall living parenchyma cells are there. Thus, in the earlier stages, very few parenchyma cells occur scattered in the xylem core from the mixed pith. The lateral central parenchymatous medullosa is developed in the medulla of xylem. The pith is surrounded by the ring of xylem, a ring of phloem, and pericycle. So, such a medullated protosteel where the xylem appear hollow to cylinder surrounded by the pith is, which is described by the Jeffrey as a siphonosteel. What is the siphonosteel? Means the centrally a medullosa or pith is there, parenchymatous pith is there. So, that is the siphonosteel. So cypress steel is again differentiated into two categories are there, the ectofluic siphonous steel and amphifluic siphonous steel. Ectofluic and amphifluic. What is ectofluic siphonous steel here? Ectofluic siphonous steel, another one is the amphifluic siphonous steel. Ectofluic siphonous steel, the xylem is cylinder enclosed, the pith is surrounded by the central palm of the only the outside the pericycle and endodermis is there. Ectofluic siphonosteel means xylem which is covered only the outside. The pericycle and endodermis and phloem is there. But here the amphifluic siphonosteel, yesterday we discussed that amphifluic siphonosteel is observed in the Marsilia. Amphifluic means both sides. So xylem is covered the both side in the inner side and outer side means phloem, pericycle and endodermis. These are there. That one we call as the amphifluic siphonosteel. Means the outer phloem is surrounded by outer pericycle and outer endodermis. The inner side, inner phloem is surrounded by the inner pericycle and endodermis. Means two pericycles are there, two endodermis are there, two phloems are there, which is covered the xylem. That one we call as the amphifluic siphonosteel, which is present in the only the Marsilia. Okay. So next one is the solonosteel. What is the solonosteel here? This is the solonosteel which is preferred by the scattered leaf gap is there, not overlapping each other in described as solonosteel. Okay. So here, what is solonosteel means? So here, so this is solonosteel. The solonosteel here preferred by the scattered leaf gap is there. So that's why there is no overlapping. This one is the solonosteel. Leaf trace is the leaf gap is present. So there is no overlapping each other. This one is the solonosteel. The solonosteel is maybe ectofluic, either amphifluic. Okay. So here, based on the presence and absence of this solonosteel here, there are the two types are there. That is the club mosses, arch steel, cyclosteel, which is forming the leaf gap. There is no leaf gap is not there. In the ferns, gymnosperms, angiosperms with the megaphilus forming the leaf gap is there. What are the leaf gaps and what not are the leaf gaps? Based on this, the structures here, um, they are to divide into different types are there. Okay. So next one is the dictyosteel. See here. Okay. The next one is the dictyostele. So here, in many of these cyclosteel pterophyta, the vertical shoot to axis is very sharp, and leaves arise in the crowd matter. This is the result of further completion of the steel, where the several leaf gaps overlap each with each other, or overlaps. Leaf gaps are present with each other. And there are the due to this, the vascular cylinder is much restricting to the uh, appear as a cylindrical meshworks with a large leaf gaps are there. Right? A cyclosteel with a more overlapping leaf gap is described as a dictyosteel. So see here, the leaf gaps are there. Means each there are each patches are a different steel. Means there are to uh, split into the number of steels are there. That one we call as a dictyosteel. Means the with a more overlapping leaf gap, number of more than two or three leaf gaps are there. So that one we call as dictyosteel. So this is dissecting the type of steel. And dictyosteel is a transverse section appear as a separate vascular strands arranged in the form of a ring. Each vascular strand, so in the form of a ring-like structure. These vascular strands separated by other is the leaf gap known as the meristeels. So that is a separate to steel we call as the meristeels. They are to separated by the leaf gap, leaf traces or leaf gap. So each meristeel is nothing but a protosteel. So dictosteel condition is entirely different from the other polysteel condition found in the Selaginella. Okay. So in certain leprosporangia, it forms in addition the leaf gap is perforation are formed in the vascular cylinder, resulting in the formation of the dictosteel. Such steel is known as dictosteel. So next one is the polycyclic steel. So here, U steel is nothing but U steel means the centrally uh, one is the pith is parenchymatous pith is there, which is surrounded by the vascular bundles are there. Okay, vascular ring like vascular bundles are there. The next one is the polycyclic steel. Polycyclic steel means here the ultimate the complication of the stellar organization in the pterophyte consisting of development a number of a separate steel one inside in other. Okay, so the independent steels are there within the steels. A number of separated steels are there inside means within the other steel. This is known as the polycyclic steel. The polycyclic steel have the pteridium. There are three in the motonia, even four in the teres. So the pteridium is the outer cylinder is the dictio steel. Have the several meristeels are in the stone steel with a two leaf gaps. So another modification of the cyclone steel found in the advanced type that is the U steel. See here, U steel. Another type is the U steel. So in this vascular consists of the collateral and bilateral vascular bundles with a leaf gaps are there. Between the leaf gaps are between the vascular. U steel is characteristic in higher plants as well as gymnosperms and dicotyledons. Okay, that is the usual condition. Vascular bundles are there, a number of leaf gaps are there. These vascular bundles in the form of a ring. So next one is the atactostele. Next one is the atactostele. Means atactosteles means this is most complex structure in the found monocotyledons only. Here consists a number of vascular bundles are scattered in the parenchymatous tissue, that is the connective ground tissue. Atactostele is commonly found in the monocots. Eustele found in the dicots, as well as the gymnosperms also. Eustele and atactostele. So what is the difference here? Eustele atactostele means in eustele vascular bundles are formed in a ring-like structure, but atactosteles are there is no form a ring-like structure. Means they are all the scattered in the connective tissue or ground tissue. This is the mainly in the monocots. Okay, this is the dictyostele, eustele, and atactostele. Another one is the polycyclic scale. Polycyclic scales means more than two cycles are means steels are present within the steel. So what is the some evidences for the stellar theory? So the stellar theories advocate the protosteel is the most primitive and is undergo through several variation leading to the evolution of the dictyostele or nothing but polycyclic steel. So here the saprophytes with the protosteel occurred in the upper Silurian and lower middle Devonian period. The study of the anatomy of sporophyte of living Carotophides has shown the recapitulation of uh, the several changes pronounced by the stellar theory. In Motonia, the Pectunia stem is pulling the beings the protosteel possessed through a brief solostelic phase, later and the monocyclic steel, finally polycyclic steel is there. So here that has these polycyclic steel is the stem apex is steel through a past birth. Okay. So here in the Beauvoir, they point out the following steps lead to the development of steels in the fern to the protosteel. What happened here? A solid xylem core and the heterogeneous xylem, but without peak, and central pith surrounded by the xylem rings. The interruption of the steel by the leaf or overlapping the leaf gaps, appearance of the internal phloem and endoderm. This is the some developmental stages are there. So they are established the polygenetic relationship among the pteridophytes. The evolution from the protosteel to siphonosteel and siphonosteel to dictyosteel as takes place in the several independent lists and in different rate and changes may be observed within a single family or even the single genus. So what is the evidence are there means what is the evolution from protosteel to siphonosteel and siphonosteel to dictyosteel. Dictyosteel is nothing but is polycyclic dictyosteel. This is the evolutionary stem. Is okay? So next one is the heterospore. What is the heterospore here? So in the heterospore means is clear? Hello? Stellar evolution is okay?
Yes,
Yes,
sir.
sir.
Okay. So here, the next one is the heterospore and seed habit. What is the heterospore means? The pterophytes are mostly homosporous condition is there, where the sporophyte produced only a single type of the spore, so that is called as the homosporous. Means are the all spores are physiologically, structurally, they are all look alike. So, so this is the homosporous condition. So, which has to germinate means they are the monoecious conditions are there. So, bearing the both male and female reproductive organs are there. But a few genera are Selaginella and Marsilia, the sporangia, are to form the two types of the spores are there. That is the microsporangia, they can produce the microspores, and megasporangia to produce the megaspore. So this is this type of the two types of the spores. The property is known as heterospore. So in the living pteridophyta, the heterosporia occur in 8 genera are there. That is the Selaginella, Ascites, Marsilia, Pulilaria, and Ringidium, Salvinia, and Azula. They can see the heterospore condition in these 8. So, in the paleobotanical and experimental and developmental evidences suggest the heterospore is the developmental due to the disintegration of the certain number of spores in the sporangium. Okay. So, as a result, the disintegration of the certain number of spores and the remaining spores that are survived in the sporangium have better nutrient and specificity naturally grows into the specialized spores are there. So on the basis of the obtained the evidence of fossils in equisitales, there are the started with the disintegration of the certain number of spores in some sporangia as redundant, possibly the excessive development and other maybe lead to the formation of megaspores. So the experimental evidences are there. What are the experimental evidences? Here they have shown nutritional factor in the responsible for the origin of the heterospore. Nutritional. So in Selaginella, the plant grow under feeble light have to produce the stabile containing only in the microsporangia because there are retained the rate of the photosynthesis. Similarly, in the Marsilia, the effect of nutrition was investigated by Chatak in 1910. The temperature was lowered by the sprinkling cold water spray in the Marsilia and was found in all the megaspores was grown under the favorable conditions. So the temperature was elevation. So the temperature was lowered by the sprinkling cold water spray. The Marsilia found only the megaspore, and the large number of microspores are present in the sporocarp. When the same plants were undergrown the favorable condition, it was found in those sporocarp where all the megaspores and large number of microspores were killed. The remaining microspores show the increased size up to 16 times in normal condition. So based on the environmental factors also means experimental evidences are there. Okay. So increase in the number was the size was obviously due to the increased supply for the food material. The largest of these spores have to reach the size of the megaspore. They attain some similar structure features are there. In homosporous form, the sex determine they accept their influence on the mega gametophytes during the formation of sex organs. But in the case of heterosporous form, the sex determination accept their influence on the spore mother cell. So the influence of the sex determination is shifted from the gametophyte to sporophyte. It is a small shift, but the significant is once it is the result in the origin of heterospory in the vascular cryptogam. So either in the Selaginella, ascites or Marsilia, all the spore mother cells are microsporangium undergoes to meiosis to form the microspores, whereas the megasporangium, but all the mother cells are died. This is the surviving spore mother cells undergoes to meiosis to form four megaspores, while only one is mature. So another is the manifestation of the heterospore. What is the manifestation of the heterospore? It means the heterospore is marked by the forming the spores in two different sizes. That is, smaller microspores and larger one is the megaspore. Okay. So here, so in microsporangium, the number of microspores produced is many, if not numerous, in the megasporangium. Limited number of megaspores are produced. The usual number of megaspores is either 4 or 1. In Selaginella, 8 megaspores are found, whereas in Selaginella, Repentis and Monospora only one megasporangia found. In the differentiation, the size of these spores is related to distinct sex gametophytes on the germination of the small microspore and male gametophyte and the megaspores are produced with the female gametophyte. So the male gametophyte bear the antheridia and the male organs, and female gametophyte bear the archegounia, which is a female organ. Thus, the heterospory has brought the unisexuality of the gametophyte, and sexuality it is controlled by the preceding the diploid generation. Okay. So here the heterospory has resulted in the reduction of the haploid and gametophyte phases, which is situated in being the independent. Derives its food material from the parental diploid phase. Okay. So the gametophyte are also reduced in the accommodation of themselves in the spore. The male gametophyte is more reduced than the female gametophyte. The vegetative body, the vegetative body of the male gametophyte is restricted to a single path of prothali cells. The female gametophyte is compared to large with the several vegetative cells. containing the large food reserve with the help of the nourish and the embryo formed after fertilization. So, so another effect of the heterospore is the retention of the megaspore with the tissues of the sporophyte until after fertilization. In collagenol, the megaspore is not liberated immediately but is retained with the megasporangium where it is developed into a female gametophyte bearing the archegonia at maturity time. The entire female gametophyte is found within the spore wall. The gametophyte of algae, bryophytes, even certain pterophytes are independent. But in the heterosporous pterophyte, the female gametophyte is dependent on the sporophyte. Okay. In the collagenol, the development of female gametophyte depend upon the food store in the megaspore, but upon the nutrition matter supplied by the tapetum of the sporangia. The collagenol has offered the several illustrations related to the origin of evolution of seed habit. After attaining such as the large step to existing the collagenol apoda to the seed habit in the primitive spermophyta. What is in the Leginopteris, the fossil pterodosperms, the Megasporangium has to protect to cover surrounding it. In the inner cover, it is enclosed the sporangia except the apex is described by the integument such as the sporangia with the integument is an oval. So during the Permian period, which is followed by the Carboniferous period, further changes may have be probably occurred in the production of the mechanism and to adapt the plant for fertilization without external water for the siphonogamy. The cycads and ginkgo have been surviving the Permian period almost unchanged. They illustrate the mode of the reproduction at the same time. The sporangia are born in the dense cones which are distinguished as the male and female sobal. Okay, so the megasporangium is provided with the protective integuments with an opening at the apex that one we call as the microspores. The megaspore formed meiosis developed into a female gametophyte that bears the two archegonia. The female gametophyte is formed entirely with the megasporangium. In the megaspores after their liberation form the microsporangia and dispersed by wind or catch in the fertilization drop into the certain secretions at the tip of the megasporangia. Some secretions are there on the tip of the megasporangia. As the fertilization is the drop dice of the megaspores are chopped into the megasporangia. After getting the microspores, they germinate to produce the pollen tube that grows involved in the megasporangium to absorb the food material. So, in the opening of the integument, which is closed, so spermatozoids are released to the closed chamber, swim to the archegonia in the liquid eggs. So, after fertilization, the oospore developed into the embryo and became the dormant. The integument surrounding the megasporangium became the hard and woody. So such a structure is considered the diploid plant, which is surrounded by the remaining case of codopetalum. The pollen that germinate from the microspore moves toward the archegonia. So here the spermatozoites travel through the pollen tube and carry down the archegonium. This method which is spermatozoites are transported by the means of pollen tube and spermatozoites. The spermatozoites had reached the that one that reproduction is called as a siphonogamy. Means pollen tubes are transported. So this adaptation saphenogam is found in the flowering plants, so it is most important final stage toward the seed habit. In the cycas, that is the cycads, the ginkgo and coniferous and other flowering plants, the megaspore is not an isolated and free structure, but is also histologically united with the megasporangium. So what is the here? After consideration of the above account, it can be concluded the Selaginella and Marsilia, which have made the several steps advanced toward the formation of the seed habit, could not be proceed further in the form of seeds because of these evidences are there. The absence of protect the structure like integments around the megasporangium and retention of the megaspore with the megasporangium has not been established permanently. The absence of histological union between the megaspore and megasporangium, the absence of dormant period of the embryo. So, Selaginella marsyria are the adapted for the saphenogamy and are developed on external water fertilization, which is the primitive step, which is direct into the seed forming habit. It can be finally concluded the heterospore leads to the seed habit. Okay. Hello. Hello. Does anybody
Yes, sir.
respond? Okay. So, what is the heterospory and what is the seed habit? So, the heterospory to leads the seed habits. Okay. So, in from the stellar evolution and heterospory and seed habit, the stellar evolution is definitely questioning this a question. Okay. Stellar evolution. They are asking yes. Yes. And another one is the heterospory and seed habit. This is also asking in your examination point of view. Okay. Okay, Nandi, clear? Huh?
Yes, sir.
So, in the what is this one here? In the stellar evolution, with the diagrams you should draw. Okay, still so you should draw these diagrams. On the textbook, also different types of the diagrams are there. Solonosteel, plectosteel, actinosteel, saprophyte, saphnosteel. Okay, like this structures you can draw. Dictyosteel and amphiphytic saphnosteel, polycyclic dictyosteel. Okay, Nandi? Okay, clear?
Yes, sir.
Hmm? Okay. So thank you for attending. The remaining only two topics are there, that is the gymnosperms and pteridophytes. Okay. Remaining two topics are more gymnosperms and another one is the uh, fossils.
Finance and Guineatum.
Huh?
So, block two important questions.
Block two?
Important questions, sir.
Block two?
Yes,
చెప్పాను
sir.
కదా నిన్న ఆఫ్టర్ క్లాస్ ఐ ఆల్రెడీ టోల్డ్. So block wise, topic wise, already told now. In block two, okay. In block two, some important questions here. The general characters of Heterophyta and the classification of Heterophyta. That is very important. Okay. Only classification. Another one is Rhinia. Structure of Rhinia and sporangia of Rhinia. That is the sporangium. That is very important. Rhinia and Lycopodium. Lycopodium morphological features and transverse section of the stem. Lycopodium, that is important in your theory and as well as practical. Lycopodium transverse section, stem, and cones. Less of cones also. Lycopodium cones. Equisetum morphological features means external features and stem. Equisetum transverse section and equisetum cones. That is very important. And another one is the Marsilia. Marsilia plant body means sporophytic morphological features. External features, nothing but. And rhizome, petiole. Marsilia rhizome and Marsilia petiole and sporocarp. This is the very important in the Marsilia. So today class is stellar evolution. This is also asking essay question. This is compulsory question. Stellar evolution in Pteridophyta. Heterospore seed habit is also sometimes they are asking as a short notes. Okay. Okay, clear.
Yes, sir.
Okay, Nandi. Okay, Andy. Thank you, Andy. Thank you for writing. The remaining two classes are there. Tomorrow and the day after tomorrow. Tomorrow we discuss the gymnosperms. Mainly important points are there. That is the pinus and the needle. Pinus cones, that is very important. Pinus needle leaf structure is also. Pinus leaves we call as the needles. Needle-like structures are there. Very pointed structures are there. That one we call as the needles. Pinus needle, transfer section, and male and female cones. Ovule is also. Next one is the netum. Netum plant body and netum ovules and chobalas we discuss that one. Okay. Okay, Nandi, clear? Huh? Hello.
Yes, sir.
Okay. Okay, Andy. Thank you, Andy. Thank you for attending the class.
Thank you, sir.
Thank you, sir.
Okay, thank you.